Welcome, everybody. We're going to get started. First and foremost, welcome to the first of our medical genetics specialty series. The goal of this specialty series is to really share our genetics expertise with different departments across the health system and different types of providers. Just a few housekeeping for this specific webinar. Please enter any questions you have in the Q&A box. We'll answer all questions at the end, but my colleague Leah Zaretsky has offered to moderate so that if a question comes up that really needs to be answered before the end, she will let me know. Also at the end, there will be a survey, and please feel free to suggest any other topics you would like to hear from us for future webinars or any specific feedback you have. And please stay tuned for future information about upcoming webinars. We already have the next two scheduled. The next one will be on July 29th about newborn screening, and then the following one will be about implications of somatic testing on germline analysis, which might be of particular interest to this group because it's also oncology related. Some of the other topics that are in the works will include mosaic embryos, as well as neurogenetics and undiagnosed disease and genetic etiology. So please stay tuned and please feel free to share any feedback. Also, I have no conflict of interest. I have no disclosures. So the goals for today's webinar is a few goals. First is to review the foundation of hereditary cancer, and then we will use that foundation to discuss the indications for testing, risk assessment, how to choose a genetic test, and interpretation of results. This will hopefully set a context for different providers to have discussions with patients, but also set a foundation for future webinars. So we might err on the side of being a little bit more basic with this webinar so that in future webinars we can build upon this one. So just a little bit of background information so we're all on the same page. When we think about hereditary cancer, that's actually a very small proportion of cancer in general. Most cancer we consider sporadic cancer, meaning happening by chance. And when we think about hereditary cancer, what we're thinking about is a specific genetic change in a gene that's supposed to be protecting against cancer that can be passed from one generation to the next that could put an individual at a higher risk for cancer. There is this overlapping region, and I often refer to this pie chart as potentially even a Venn diagram, where we see familial cancer. That's when you'll notice that a family has more cancer than you'd expect. And it could be due to small genetic factors that we just can't detect at this point in time, but also environmental factors where families grow up together, have similar exposures, have similar eating habits, exercise habits, and therefore the environment might be shared. But we'll be focusing on hereditary cancer. Also, this presentation will be sent to everyone after the talk. If you have any technical issues, please chat Jennifer Gutierrez. She will help you as well. So when we think about sporadic versus hereditary cancer, just the molecular background, in sporadic cancer, we would think about someone having two working copies of every gene, and over time, they accumulate a mutation, but they still have that second copy and a second layer of defense. But over time, they accumulate a second mutation, and that's when a tumor can develop. This is known as the two-hit hypothesis. But with hereditary cancer, that individual is born with one non-working copy because they have a mutation in that gene. And that is most of the time inherited from a parent, either mother or father. And so we kind of describe this as being one step closer to developing cancer. So that doesn't mean that this individual will ever develop cancer, but they're one step closer. I also want to point out that this isn't true for all of the hereditary cancer genes, but this is for many of them. Just some definitions and backgrounds. When I use the word variant means a change in the genetic sequence that's different from the reference. This can be due to, this is a neutral term. So a variant just means a change. It does not mean that it causes the gene not to function properly, but more and more people are using the term variant to connote a mutation or a pathogenic variant to connote a change that does cause the gene not to function properly, but it actually is a neutral term. The word penetrance connotes what is the likelihood that a clinical condition will occur when a particular genotype or when we find a specific genetic finding. So a lot of these cancer genes, they don't have full penetrance, meaning that just because someone has a change in these genes doesn't mean that they will definitely develop cancer. And so we describe penetrance as the risk for someone to develop cancer. And this can be organ dependent, sex dependent, or age dependent. So for example, we might describe what is someone's risk to develop cancer by 20, 30, 40, 50, and those are age dependent penetrance. So let's get into the meat of things. Indications for referral. So before a patient even comes into our office, what are reasons why we might see them? And I like to call these the red flags of hereditary cancer. And this is really largely dependent and explained by that two-hit hypothesis. So when we think about red flags, we think about a young age of diagnosis. So again, if someone is one step closer to developing cancer, it would make sense that they are at a younger age when they develop cancer. And we think about this as under 50 for colorectal or endometrial cancer, under 45 for breast cancer. We also think about rare cancers, so that could be ovarian, pancreatic, male breast, metastatic prostate cancers, pheochromocytomas, paragangliomas. And so this is important to know because if someone has a family history or a personal history of any of these rare tumors, they are automatically qualified for genetic counseling and genetic testing. Certain pathologies make a more common cancer into a more rare cancer. So for example, triple negative breast cancer, though breast cancer in general is more common, triple negative breast cancer would be considered more rare. We also think about multiple cancers. So again, if there's a hereditary component, we would expect that this genetic change is in all the cells of the body, and therefore the same person would have an increased risk for multiple cancers. And then if it's passed on in the family, we would expect to see multiple family members on the same side of the family. And then there are some non-specific to cancer, but other findings we might find that would put someone at a higher risk to have a hereditary predisposition. And that could be an ethnic background, but also other associated findings. Sometimes we get referrals for certain skin lesions that aren't cancerous, but could be an indication of a specific hereditary predisposition to cancer. When we evaluate the indication, it's really important for us or other providers to know why is the patient coming in now. And so sometimes patients come in because of a personal history, and so that could be due to a recent diagnosis, and that can help the patient make a surgical decision or a treatment-related decision. It could be a family history, so someone who's unaffected but is trying to determine what their management should be going forward based on their family history. And two more newer reasons for referral include direct-to-consumer testing, which we'll talk about more, and reproductive considerations, which we'll also talk about more. One point I want to make in terms of indications is that there has been a big expansion of criteria for genetic testing. And this is partially due to the fact that we're learning more about how these hereditary predispositions present. So for example, we now know about the association with BRCA1 and 2 with pancreatic cancer or prostate cancer. And so that's included in the criteria, but also the ability to test for multiple genes at the same time. And this is from the National Comprehensive Cancer Network. And my pointer wasn't working before, so hopefully you can see this, but we actually see that they've changed their criteria. It's no longer hereditary breast and ovarian cancer criteria, but high penetrance breast and ovarian cancer susceptibility genes criteria to account for the multiple different genes that we're able to test for at the same time. And it has gotten much longer because the criteria has expanded. The one limitation to this is unfortunately not all insurance companies have followed NCCN. And so just because they meet NCCN criteria, not every insurance company has followed this expansion. Let's talk about direct-to-consumer testing. So in 2018, the FDA authorized 23andMe to report out 3 specific Ashkenazi Jewish founder mutations in BRCA1 and 2. And then in 2019, they authorized 23andMe to report out 2 sites in the MUT-YH gene. For those who aren't familiar, the MUT-YH gene is associated with recessive polyposis, meaning that someone has to have 2 non-working copies of the gene to be at risk to have a polyposis, though having 1 not working copy of the gene does slightly increase the risk for colon cancer. And MUT-YH, these specific MUT-YH mutations are extremely common in Northern Europeans, about 1 to 2% of the population. But a couple limitations that you should know about with 23andMe. First is, this is from 23andMe's own package insert, that any test that's done through 23andMe has to be confirmed by diagnostic testing. And therefore, before any treatment decisions are made for a patient, they need confirmatory testing. The other factor that we have to think about is that when a patient says that they're BRCA1 and 2 negative through 23andMe, that means that they were really only tested for the 3 common Ashkenazi Jewish mutations. And so if they have a strong family history of breast, ovarian, pancreatic, prostate cancer, that probably is not enough. And even more so if they're non-Ashkenazi Jewish, it really is not very helpful for them. And so there's a sense where that 23andMe testing is actually causing false negatives. So a patient thinks they're negative, but really they haven't had a comprehensive test. The second factor that we have to think about is that even though 23andMe is not allowed to report for other areas of these genes or other hereditary cancer genes, what they do allow is for patients to download their raw data, and then they're able to upload this data into other sites. And what this causes is false positive and false negatives. Because this data does not have quality control, and so it's not cleaned up, and also these third-party sites are not clinically certified to evaluate the data. Sometimes we've had patients who will come in and think that they're BRCA positive, have Lynch syndrome, have a polyposis syndrome, but it was a false positive. This was a study put out that's found that 40% of variants were actually false positives. So it's really important when a patient tells you that they've had a positive genetic test to get the report and understand where they're getting this information. Many of us are providers in New York State, and New York State has specific laws actually regulating genetic testing labs, that the genetic test and the lab has to be New York State approved. The other indication that's newer is reproductive implications, and I had seen that a lot of OBGYNs signed up for this webinar. So this is specifically important for these groups, that some patients are also coming to see us before having children or during a pregnancy. And so this could be because the hereditary cancer gene is associated with a childhood onset condition, but more and more we're seeing it's due to recessive conditions. And that's because some of these genes associated with a dominant risk, meaning having 1 not working copy, is enough to have the condition. When it's inherited in a recessive manner, causes a different condition. So for example, Lynch syndrome, if one parent has Lynch syndrome and the other parent has Lynch syndrome caused by the same gene, there would be a 1 in 4 chance that they both pass down the not working copy of the gene and have a child with a condition called constitutional mismatch repair deficiency, which is a childhood onset cancer predisposition condition. BRCA2, if a child inherits 2 non-functional BRCA2 genes, they would have a risk for a condition called Fanconi anemia, which is a bone marrow failure condition. And so what we're starting to see is that patients with these adult onset conditions are coming to see us and testing their partners because of the recessive conditions associated. The other reason that we're seeing more reproductive patients is also because of carrier screening. So historically, we thought of carrier screening as testing during pregnancy or before pregnancy for conditions that we would expect the individual not to show any signs or symptoms, and then only if both members of a couple are both carriers that they would have that 1 in 4 risk to have a child affected with these conditions. But that model isn't complete because now we're seeing that some of these genes, carriers do have risks. And so for example, ATM, which is associated with the autosomal recessive ataxia-telangiectasia, we actually know that women with ATM mutations are at an increased risk for breast cancer and men and women for pancreatic cancer. This breast cancer link was actually first discovered because we saw that the mothers and grandmothers of children with ataxia-telangiectasia were developing breast cancer at a higher rate. And so now these individuals are undergoing carrier screening as related to their pregnancy, but finding out that they're at an increased risk for cancer. This is especially important in the case of ATM because sometimes we find out this risk at an age when we wouldn't actually start screening. And so finding out this information at that time could be a little bit more sensitive. The other factor is with the expansion of IVF, especially with pre-implantation genetic testing for monogenic disorders, so being able to test embryos. Some individuals, when they have a strong family history of cancer and they are thinking about undergoing IVF anyways, would like this information to have the option to potentially test embryos. With these patients, it's also very sensitive because a lot of these patients want to start the IVF process as soon as possible, but hereditary cancer testing can take 2 to 4 weeks to come back. And then if we do find something, to create the probes to be able to test embryos can take a much longer time. So understanding the timeline is important for these patients. Once the patients are even in the door, we conduct a risk assessment, and this is based on gathering detailed information about family and medical history. This starts out with a conversation about who is the most informative person to be tested, and I'll talk about that more in a second. But we're taking into account all these factors, these red flags that I was talking about before, for us to determine what is the likelihood that this is hereditary or environmental. And then also, can we figure out what genes might be associated that could explain this history? And then also, what are the limitations in this history? And this is a really important point because sometimes a patient will say, well, I really don't have a strong family history of cancer, but if their mother and grandmother and sisters and aunts all had hysterectomies for fibroids, then that could really be obscuring a family history of uterine cancer. If there are a lot of men in the family and not a lot of women in the family, then that could be obscuring gynecologic or breast cancer risk. And then some families are just very small. If everyone's an only child, there's not really that many relatives to have developed cancer. When we talk about the best person to be tested, I just want to give a case example because I think this is a really important point. This is a case that I saw a few years ago, and this is when we take a family history, we draw out these charts. They're called pedigrees, where women are circles, men are squares. And people in the same generation are on the same line. So for example, my patient is a 61-year-old female. Her sister had breast cancer at 34 and is now 58, and she came because her other sister had been recently diagnosed with a triple negative breast cancer at 61. Their mother had been diagnosed with breast cancer at 34, and her father had been diagnosed with colon cancer at 50. And she had this, her mother's first cousin had been diagnosed with ovarian cancer and passed away of the ovarian cancer in her 40s. And this is another example of what I was talking about, men in a family being a limitation when it comes to assessment for breast and gynecologic cancers. So while this ovarian cancer is a little bit distant from her in terms of relationship, it's through 2 men that are unaffected and then only through affected women. So I think that's an important point. But when my patient came to me, spoiler, she tested negative for everything I looked at. She had been tested, her 2 nieces who hadn't had cancer were tested, and her sister who had the breast cancer at 34 had been tested, and they all tested negative. But when we look at her mother and her other sister, they had not had testing, and the woman who passed away from the ovarian cancer also didn't have testing. And so when I look at this family tree, I'm left with the question, is there something going on? Because this is a very strong family history that we just can't explain, and that maybe technology can't pick up. Or is there a mutation in this family that her mother and sister have that they just weren't tested yet? And so we had this lengthy conversation, and she encouraged both her mother and sister to go for genetic testing, and they actually were BRCA1 positive. So now when I do a risk assessment for this family, it's a very different conversation. Instead of thinking about, 3 first-degree relatives with breast cancer, I can say 2 of these breast cancers were really related to the BRCA1 mutation, and I would guess the ovarian cancer was also related to this BRCA1 mutation. And so now we're only accounting for the breast cancer in her sister that's still unexplained. And actually, when we run risk models before her mother and sister were tested, she qualifies for a breast MRI. And when we run risk models with the knowledge that there's a familial mutation that she didn't inherit, she actually no longer qualifies for an MRI. So that's a very big difference in terms of management. We did do a multi-gene panel for this patient because of her father's colon cancer at 50, which was also negative. Once the patient is in and wants testing, we do have to choose which test to perform. And I think this is a good point to stop and say, does the patient even want testing? And there are lots of different reasons why patients don't want to have genetic testing. One that we often get questions about is insurance discrimination. So there is a law called the Genetic Information Non-Discrimination Act, which says that health insurance cannot discriminate based on genetic testing results. So there are some loopholes to that law as well, but this exempts life insurance, long-term care, disability insurances. So I've had many patients who have said, I don't want testing right now, but I'll come back when I have my life insurance in place. But that's just one of many reasons why patients do not want testing, and we do respect a patient who decides that this isn't something for them. But if they do want testing, the question is what test is the best test for them? And that's really dependent on the indication for testing, and then also determining based on this indication, what are the different laboratories' capabilities and technologies? So when it comes to an indication for testing, why does the patient need this information? So if they're making a surgical decision, and this is from a brochure from Invitae Genetics, we would probably order for a surgical decision a stat panel that would come back sooner, which would have genes relevant to their surgical decision. If it's treatment related, well, what type of treatment are they thinking about? Are there certain genes that if we test for could potentially get the patient on a clinical trial? Is this for family members? And then we might want a broader panel. But also, what type of information does the patient want? The reason why I like this brochure from Invitae, even though they've actually discontinued making it, is that if you see the genes in black, they call it their guidelines-based panel. What that means is those are genes that we have enough evidence to make specific recommendations about what the patient should do based on the findings. The extra genes in white are genes that we don't have enough evidence, or the risk for cancer is not high enough that the medical community has made recommendations about what to do about finding changes in these genes. So some patients don't want information that they can't act on. So having that nuanced conversation is very important. And then the other question is, if the indication is for a specific condition, does the lab even have testing available? So for example, testing for hematologic malignancy in a family is a newer emerging hereditary cancer predisposition test that we can perform. And so not all labs have heme panels. The other factor is that, for example, one example is RNF43 is a gene associated with sessile serrated polyposis. That's a newer gene, but not every lab has it. So if a patient is coming with sessile serrated polyposis, I'm going to send to a lab that's able to test for that. The other question is lab capability and technology. So first and foremost, if there is a familial mutation, where was the relative tested? Because labs have such differing capabilities and technologies, we want to ensure that whatever was picked up in the relative would be picked up in the patient. And then how does the lab evaluate results, which I'll talk in a little bit more detail, and then technological capabilities, which we'll also touch upon. When we talk about how labs evaluate results, one of our big questions are about how do they interpret whether a variant is pathogenic, meaning that it is disease-causing, or benign, meaning it's just what makes one person different from the next. And this is largely based on guidelines by the American College of Medical Genetics and Genomics and the Association of Molecular Pathology. These guidelines are based on multiple different types of evidence. So it's much more complex than we typically think about. It's based on population data, computer data, functional data, and a lot of other pieces of information. I think what's really important also to note is that we don't only need evidence to prove that a variant is pathogenic, but we also need evidence to prove that something is benign. So if we don't have enough evidence to say something is benign, we leave it in this third category called a variant of uncertain significance. So with any genetic test, as it relates to hereditary cancer, we have three possible results. The first is a pathogenic result, or likely pathogenic, that's positive. The second is negative, likely benign or benign. And then if we don't have enough information to put it in either of those two categories, it gets put in this variant of uncertain significance, which anyone who's seen genetic tests as it relates to hereditary cancer have probably come across these. And that's because they're so common. This is a visual aid provided by Ambry Genetics about some of their panels. The arrows are pointing specifically to a panel related to breast and gynecologic cancer genes. What you'll see here with the red arrow is about 10% of patients test positive. 63 or up to 64% of patients will test negative with that panel, but 24% have a variant of uncertain significance. So that's extremely high and extremely common. What do we do about these variants? So we do our homework. The first is we look into it. This is a screenshot from a database called ClinVar, where labs can upload their data and their interpretation about specific variants. Not all labs do share their data. And this allows us to compare if maybe a different lab has differing information about the variant. So in this case, all the labs are agreeing that it's of uncertain significance. Whereas sometimes, though, not all labs agree. And I specifically chose this variant because I knew that there's conflicting evidence, where most labs are calling this pathogenic or likely pathogenic, but some labs call it uncertain significance. The reason why I chose this particular variant and the reason why there's controversy over it is that this particular change in the CHEK2 gene is a Ashkenazi Jewish founder mutation. So it's extremely common in the Ashkenazi Jewish population. And this is where understanding how your lab interprets variants is extremely important. So Myriad Genetics, for example, calls this variant of uncertain significance because their databases and their variant curation is largely dependent on population data. And so for that reason, because this is so common in the Ashkenazi Jewish population, they consider this variant of uncertain significance because they think it's too common. Whereas other labs which don't weigh population databases as much, they consider it pathogenic or likely pathogenic. So it's really important to understand why is your lab calling these variants the way they are and what's the logic behind how they do variant curation. In general, though, when we get a variant of uncertain significance, we treat them as innocent until proven guilty. And the reason for that, and this is specific for hereditary cancer, is because this is from a study which looked at variant curation and variants reclassified over a 10-year period. And what you'll see in the red box is 97 to 98% of the time, when these variants were reclassified, they were reclassified as benign or likely benign. And it's for that reason that we treat them as innocent until proven guilty. So we don't make any clinical recommendations based on variants. We obviously do our homework to double check, but we don't make any clinical recommendations, and we also don't test family members based on variants of uncertain significance. Okay, let's talk about technology, because this is, I think, some of the most exciting aspects, is that technology in genetic testing and understanding of genetics is rapidly developing. And overall, the main goals of developments in technology is to increase the diagnostic yield in genetic tests to pick up patients and be able to find the hereditary predisposition in families that we couldn't previously identify, while also reducing the variant rate. And so this is a timeline provided by Ambry Genetics. If you see, it starts in 2006, which is actually very recently, with deletion, duplication analysis, and rearrangement analysis. And the way I explain that advancement is, if you think of spell check, that's looking for spelling mistakes, but you won't pick up if a whole sentence or a whole paragraph is missing. And so that was one development. Then with next-gen sequencing and panel testing, we're able to test multiple genes at the same time. And then I'll talk about what RNA analysis is in a minute. But just to give you an example of how these developments really impact families, this is a patient I saw 2 years ago. So his mother was diagnosed with breast cancer at 44, and then diagnosed with ovarian cancer at 56 and passed away from the ovarian cancer at 59. His sister had originally had genetic testing in 2007, just as deletion, duplication, rearrangement testing was coming out, but not fully available, and she was negative. Her doctor then in 2018 ordered BRCA1 testing and actually a full panel, and she came back positive. This is from an outside provider, and the outside provider is who wrote the exclamation point on the report. And that's because they were really shocked. They thought, well, she's BRCA1 positive, we're not going to find anything in those genes. But DEL exons 21 through 24 shows that this is a deletion, and that wasn't, the tech before couldn't pick up deletions. So suddenly she was positive, and she took risk-reducing management decisions. So she actually had a prophylactic mastectomy and an oophorectomy and salpingo, and her fallopian tubes removed. And then her brother, who is my patient, he got tested and was positive. And he's since had breast exams and prostate exams, and might be enrolled in pancreatic cancer screening studies. But this was 2 years ago. Recently, his daughter, who's now 24, going to be turning 25, has come, and her testing is actually pending. So by his sister getting tested, now we're able to do cascade testing and testing other family members. So if his daughter does test positive, we will be able to monitor her appropriately. But I do want to talk about a newer development, the RNA testing. So another case example, I had a patient a few years ago. She was 61. She had been diagnosed with ovarian cancer, and she had just finished treatment, and she was referred because her doctor was thinking about using PARP inhibitors as maintenance therapy. Her mother had been diagnosed with breast cancer in her 40s, and at that time had a bilateral mastectomy and actually an oophorectomy at that time as well. And then her grandmother had died of a gynecologic cancer, but she wasn't sure what. This is another example of many men in a family. So we see all the women are affected, but none of the men are. When I did testing for her, she was completely negative at that time. Just to take us back to some biology basics, when we look at genes, we see that they're built of what we call exons and introns. So an exon is what ultimately gets, and genes code for proteins. So along the way, the RNA is the messenger that takes the message from the gene to the protein. And along the way, the intron part of the gene is spliced out or cut out. So the thought is that the RNA would be only made up of the exons. Why this is important is that a lot of genetic testing companies don't sequence or test the introns. And that's mostly because there's a lot of information that they see in introns that wouldn't be important. And so it could increase variant rate and also increase turnaround time. They will sequence a little bit into the intron because that's when they think it would affect splicing or that cutting out process. But what Ambry Genetics has recently launched is testing RNA and DNA at the same time. And the goal of this, and this is from a paper that they published, is to be able to pick up if there's any changes in the splicing that then causes exons to be skipped or introns to be included or pieces of exons to be skipped. This is just a picture of some of those examples. And so what they're doing is they're sequencing the DNA and also looking at the RNA. And if they find any RNA abnormalities that they didn't pick up in the DNA, they go back and look at specific areas of the intron. This is from this same paper, and I'll walk you through this. So what this is showing is RNA analysis on 2 negative individuals. So what is circled represents the exons. These arrows represent the introns. So what we would expect to see is exon, where you see the arrow representing the loop of the intron, exon, loop, loop being the intron, and then exon. But what we see on the left side is half of it being wild type, so seeing that exon, intron, exon, intron, exon, and then half just being exon, intron, exon, so skipping of an exon. And that's because of a splice site, basically because of a change within the intron, this exon is now being skipped. And what's the reason why it's half and half is because we'd expect the person to have one working copy of the gene and one not working copy of the gene. And this is specifically a BRCA1 variant. And why I chose this one to present is because, back to the case example, when my patient, 2 years later, I got a call from Ambry about my patient that they were reissuing the report because she had a very similar change, which would do the same thing to the mRNA and therefore do the same to the protein. And so suddenly she went from negative to BRCA1 positive. And again, the effects for this patient is now she can be considered for PARP inhibitors, as well as the fact that family members like her daughter was able to be tested and her daughter actually tested negative. This is from just Ambry Genetics also released what they're believing to be the increase in diagnostic yields based on RNA testing and the decrease in variant rate. I do want to point out Ambry is not the only ones doing RNA testing. They're just the only ones doing it prospectively, meaning at the time of blood draw, as opposed to Invitae or Myriad. Both allow for RNA testing if they find a variant to investigate further. Other labs are using other technological advances. So for example, Invitae has acquired a artificial intelligence company which helps them with modeling. And so all of these are to hope to reduce variant rate, but also increase diagnostic yield. Finally, the last factor and the most unfortunate factor when choosing lab is insurance. So whether this is in network with a patient, what's their deductible, what's the financial aid policy for the lab, how are patients notified of bills, and what are the self-pay prices. All of this is also to be discussed with a patient. And then the last step is the results. So when evaluating a result, the first question is, does a variant affect the gene function, which we've already discussed? If it does, to what extent does this specific gene or variant contribute to cancer risk? And so, especially with the ability to test multiple genes at the same time, we're actually learning different types of risk information. So there are high-risk genes like BRCA1 and 2, which I keep using as an example, which we would quote, let's say, a 45 to 85% lifetime risk for breast cancer. But some other genes are more moderate risk. So for example, ATM, we might quote a 33 to 38% lifetime risk, or CHEK2, a 23 to 48% lifetime risk. And so that's a very different conversation. And it's not only a different conversation in terms of the patient's decisions, in terms of their surgical decisions or their management and surveillance, but it's also a question of how does this explain the family history? So if we have an extremely strong family history and we find a moderate risk gene, does this really explain what's going on? And does a true negative exist? So if I test a family member and they're negative, are they really negative? And just to give you an example of this is a patient I saw who's 32 but had been diagnosed with colon cancer in his 30s or at 30. He has a CHEK2 change, which is associated with about a 7 to 9.5% lifetime risk for colon cancer. But would that really explain why he developed colon cancer at 30? I'm not sure that that would be the full picture. It's probably a contributor. And therefore, his 33-year-old sister, if she tested negative, would we really feel comfortable not recommending extra screening to her? And so this is where the moderate penetrance genes really make interpreting family histories a little bit more muddy. But it doesn't end there. It's not only about the specific genes, but even specific changes in the gene could have different penetrance or different risks. So going back to CHEK2, what you'll see here is in B and D, that is showing breast cancer with a CHEK2 mutation called 1100 del-C. That's a truncating mutation which causes the protein to end early, and then it actually gets degraded in the cell. And so you'll see there's none of the protein being expressed in B and D. That's why it looks there. In C, there is protein expressed, and that's because that's a different change that causes a change to the amino acids, but it's not being degraded. We think that the protein is probably less functional than the wild type protein, and that's why there's an increased risk for cancer, but the risk for cancer is not the same between the two variants. And so risk assessment really does not only depend on the gene, but also the mutation. And we see that guidelines that follow this. So this is a screenshot from NCCN about the recommendations for CHEK2. And if you see the comment, it says the risk for missense variants are unclear, but for some pathogenic, likely pathogenic variants such as I157T, which is what's being shown in the paper I just showed you, the risk for breast cancer appears to be lower. Management should be based on best estimates of cancer risk for the specific pathogenic, likely pathogenic variant. So it's a really nuanced approach. It's not only about the gene, but it's also about the specific mutation. Finally, does this finding explain the family history or personal history, or is this just an incidental finding? Especially when we're testing for multiple genes at the same time, sometimes we find changes in genes that really have nothing to do with the personal or family history. An example of this is my favorite incidental finding, I1307K in the APC gene. So APC, as you're probably aware, is associated with familial adenomatous polyposis. But this specific change is extremely common in Ashkenazi Jews. About 10% of Ashkenazi Jews will have this change, and it creates a hypermutable area in the APC gene. So the way I explain this to patients sometimes is that this isn't the first hit, it just gets you closer to the first hit. And so therefore we do see a slightly higher risk for colon cancer in these individuals, but it is not FAP. And so if we find this in a family history of uterine cancer, it's likely unrelated. It's more related to their ethnic background, though their colonoscopy regimen might be slightly different. So how can we help? So far we've talked about the indications for testing, risk assessment, choices of tests, interpretation of results, and all of this is really the goal is to provide the best information, but also the best care and support to our patients and their families. And so part of the goal of these webinars is also to create a connection that we're here to help. And I just want to thank everyone who attended. I'd like to thank Jennifer for her help, but I'd also like to thank my team. I'm one of the members of the Cancer Genetic Counseling Program. Our names are listed here. Please feel free to reach out to us if you have any questions. Patients can call and schedule an appointment. We will be having a provider line soon that's specific just to providers. And also, especially with the expansion of COVID, our telehealth options have expanded, and we've started these telegenetics programs where providers can set up specific relationships with us to provide telegenetics and even on-site telegenetics hubs. And so if that's something you're interested in, please feel free to reach out to Latasha McGriff, our office manager. I want to thank you again for attending our first webinar. We are really open to suggestions about how to improve this program, and please stay tuned for additional information about future webinars. Thank you so much for coming.